Shall I start sharing my slide now or wait?
I'll go ahead and maybe wait for Val to introduce you, and then we'll have you share
Okay.
if that's all right.
Yeah, sounds good.
Okay, Val, you are good to go. Oh,
Can you hear me all right?
There we go. Yep, you're good.
All right. Well, welcome back, everybody who came to yesterday's talk. The Ecology Center at Utah State University is really excited to welcome Dr. Tadashi Fukami, Associate Professor of Biology at Stanford University. He earned his bachelor's degree at Waseda University and his master's at the University of Tokyo, and his career has since taken him all over the world. He earned his PhD from the University of Tennessee, Knoxville, and then completed a postdoctoral fellowship with Land Care Research in New Zealand and started his first faculty position at the University of Hawaii at Manoa. In 2008, Dr. Fukami joined the faculty at Stanford University, where he continues to teach and conduct research today. Through his research on historical contingency and community assembly and species coexistence, he has earned the Outstanding Paper in Theoretical Ecology Award from the Ecological Society of America and the American Society of Naturalists Presidential Award. So thank you for joining us today. I'm really looking forward to this talk on nectar microbes. Super stoked. So thank you.
Thank you, Val. Let me now share my screen, the slide, if that's okay. Okay, is it working? My audio and also the slide?
Yeah.
Okay, great. Yeah, I wanted to thank everyone for the invitation. I hope my first talk yesterday was interesting for those of you who came. And today I would like to kind of continue with this theme of historical contingency, ecological community assembly. But unlike yesterday, where I talked about generally the background and current active area of research in this field. Today, I want to focus on this particular study system, nectar microbial communities, to show you what we've been doing over the past decade or so in this within this framework of historical contingency, and show you kind of what approaches can be done to study historical contingency which seems like a difficult thing to tackle. And so before I get to any of the specific results, I wanted to first make sure to acknowledge people who are involved in this nectar microbial research that I've been doing at Stanford over about 10 years now. And so these are some of my some of them that are shown here are PhD students that I advised, and some of them came as a visiting PhD student. For example, Caroline Tucker and Tess Granger were a PhD student from University of Toronto. They came as a visiting student to do an experiment that turned out to be an important component of our research. And then we have postdocs, former postdocs, and visiting researchers from few of them from Japan. And so the work that I'm going to do, as if you are faculty, you know that you can't do research really by yourself. And in my case, my role becoming smaller and smaller because there are so many other much better people who do work, and I'm kind of going along with them. And so I wanted to acknowledge that their contribution first. And then I want to step back a little bit and to get to what I was talking about yesterday. And so that I want to make this approachable, this talk accessible, even those of you who might not, who didn't come yesterday. And so just to starting from the general introduction, as a community ecologist, the common way of introducing your work is to start from the theory of biogeography by MacArthur and Wilson. 1967, and so it's been a while, but still kind of relevant concept even today. As most of you, if not all of you know, this idea is that if you wanted to understand species diversity or species composition in any local community, it can be island or any kind of local community, you have to know immigration rate, how quickly species are coming from species pool, and then extinction rate, how quickly species are going extinct from the your local habitat. And if you know those two, then you have you should be able to make good prediction, reasonable prediction about species diversity in your local community. So a lot of study research even today based on this concept. And so just as one example, there was a paper, a really nice paper published this year showing that if you go to different oceanic islands across the world and then look at the bird species diversity, you can actually use this concept to explain the avian diversity. But then yesterday I started to talk about historical contingency. And so some other community ecologists are starting to think more about not just immigration rate, the rate of immigration, but also history, more detailed history of immigration, because even if you have the same physical and chemical conditions, just for the sake of argument, that are shared by local habitats. It can be different islands, but almost the same physical and chemical environment. Even in that case, and even if you have the same same species, set of species in a regional species pool, initially theoretically mostly, but more empirical work is showing that just by having some variation in migration history, so that one habitat has particular species by chance coming early to this particular habitat, and other places they have different history of species immigration. Even just by having this variation, you can end up with different species diversity and composition. And that's what I'm going to refer to as historically contingent assembly. And as I was saying yesterday, this happens because of what we often call priority effects, which is a simple concept, I think, basically saying that any early arriving species that might come mostly just by chance, they happen to be the early colonizer, they can change the environment in a subtle way, or sometimes dramatic way, but often subtle way in the local habitat. Because they change the habitat in subtle way, the species that come later are going to be different in terms of how successful they can be in population because of this changes in local interactions like competition, predation, mutualism, and so on. And so, as I was saying yesterday, I think the good question to ask in relation to priority effects could be this question: when should history matter? Once again, as I was saying yesterday, it's often very difficult, if not impossible, to explain specific community if history is important, because simply because it's just not possible to go back in time to get the data that you need in terms of immigration history to explain what we see today. So even if that's difficult, if we can have some idea of when history is expected to be important and when history priority effects are not important and weak, then that could give us some good way of having at least some understanding of community assembly, particularly beta diversity, when you have some variation in species composition among local communities that can contribute to beta diversity among community variation in species composition. And also, as I was saying yesterday, this question can be relevant also from the applied perspective, because ecological restoration might involve sequential reintroduction of species, and also for medical and agricultural purposes, we might want to manage, for example, microbial communities. And so this question is kind of an overarching question that I would like to use now to talk about nectar microbes. So this is the general framework that I will use to talk about different results that I wanted to share with you with the nectar microbes. And so when I moved to Stanford in 2008, as Val was saying, I wanted to start some new study system, and this is a system that I decided to do. I didn't know anything about these microbes. I didn't even know that there are any microbes in the floral nectar, but somebody told me that this could be a good system, and it turned out to be really a productive system, at least from my own interest in priority effects. So we have a local study system with a Mimulus aurantiacus, monkey flower that are native to California. It doesn't really matter which flower you use, and whenever you have nectar in flowers, there are microbes inside, it turned out. And so there are fungi like yeast that are specialized to live in nectar, and we also see some species of bacteria that are in nectar. And there are several advantages of using this particular system, even though it's kind of a strange system when you think about it as a community ecology material. But there are some reasons why this system is useful for community assembly research. One is that just like any other microbial system, because of the short generation time and rapid growth, it's possible to observe community succession and assembly over many generations of the species involved. And so if you want to know historical contingency, then being able to look at communities over long term is convenient. And that's the same for any microbial system. But unlike most other microbial systems, what's unique about this system is that it's simple in the sense that we don't have too many species living in there. And so the microbial communities, many microbial communities are fascinating because they're so partly because they're so diverse. If you look at soil, there's so many species, but at the same time, even though it's interesting, at the same time, it just gets a little bit overwhelming because you don't know where to start if you want to tease apart which species are interacting with which other species. But in this case, we think that it's because of a lot of sugar, the carbon involved in nectar that leads to high osmotic pressure. The sugar can be a good resource, but at the same, if you have too much of that, the osmotic pressure can be a severe challenge to most microbes if you're not adapted to living in this particular environment. And because of that, presumably only a handful of species of yeast and bacteria evolved to be able to live there. And so we have just a small number of species, and so it becomes possible to focus on one species at a time and then species pair and begin to understand what happens overall in this community assembly. Another thing is that when in terms of nitrogen, we have a little bit of amino acids in nectar that the microbes can use, but that's very limited. And so we think it's not proven yet, but we think that that's potentially another reason why we don't see many species. There are only just a handful of species of bacteria and yeast that can reproduce and grow in number with with a very limited amount of nitrogen. Another reason why this system is convenient is that it just turned out to be that way. But many species, of course, not all species, but many of at least the dominant species, common species that we see are easy to culture and keep in the lab. And so you can get the nectar from the field and then do usual microbiology technique to plate them, and then you get the colonies, and then you can store them in the freezer, and then use those as you in whatever way you like to do community assembly experiments, which is not always the case with any most microbial systems. And then another reason is that this presents a well-defined habitat. And so the microbial species that we see in nectar, we don't see them so much in any other place except on pollinators that come to flowers. And so when you think about it, these flowers are really a well-defined habitat. And so any community ecology research, the first hurdle that you have is that it's often just you're not sure how to define spatially your communities, where your communities start and where your community end, because it's all continuous, the habitat. But in this case, because these flowers serve as habitat islands, you can focus on community assembly questions without having to think so much about how you actually make boundaries. And so that's another convenient system. Another thing is that, so microbes might be convenient, but if you are interested in immigration and dispersal and community assembly, that becomes quickly becomes a difficult thing, because you can't see where microbes are. They're so small. But in this case, the dispersal seems to happen through pollinators. And so I don't know if how well this video is showing, but in our system, hummingbirds are the main pollinators. But when hummingbirds come to old flower, then that flower might have a lot of microbes growing in nectar. And when hummingbirds then go next to the newly opened flower with the fresh nectar, which is often microbe-free, then the hummingbirds carry the species that are in the old flower to a new flower. And that seems to be how mostly the dispersal happens. Some of the species of microbes that we see in nectar seems to be able to go from flower to flower by themselves, but that seems limited. And so it's so that's why I see that this last reason is another thing that's convenient about this system. And so what I wanted to do is to tell you several experiments that we've done within this system that's all related to priority effects. Some very directly, some not so directly, indirectly, but to give you some flavor of what you can do experimentally to ask going back to that overarching question, to ask this question, when history should matter. And so first experiment that I wanted to briefly talk about involves 4 species of microbes. These are 2 species of yeast and 2 species of bacteria. These are species that we have isolated from the nectar of the monkey flower plants. And these are some of the commonly seen species. And we could have done this experiment with a real flower in the field, but that was a little bit challenging. So just to start with, we did a little bit more controlled experiments in the lab. And so instead of having hummingbirds, we had pipette and we had a PCR tube with nectar in it, and to do the experiment to see what happens. And so in one treatment, we had 2 species of yeast introduced, and then we waited for 2 days to introduce these bacterial species and see what happened after 4 days, just a simple community construction. And then, by the way, these 2 days seems to be reasonable when you think about hummingbirds. We can actually estimate in the field how frequently hummingbirds are coming to each flower, and then having this 2-day interval seems like a long time, but that seems within what's possible in the field. And then each flower, in our case, lasts for about a week. And so 4 days, looking at population dynamics for 4 days seems like it's also relevant, broadly speaking, to real flowers that we have in the field. And to look at the effect of species immigration history order, we just simply flipped the order to introduce bacteria first in another treatment, and then yeast. And we also had all species introduced at the same time. We didn't really know what exactly to expect, but we just thought that we would do this and see what happens. And so this is the results that we had published some time ago, but I thought this would be a good first experiment to show you. And so we have 4-day experiment, but it was we had a transfer for some time, and so we had the PCR tube with microbes growing, and we took a little bit of that to move to a new tube with a fresh nectar, and then we continued for about a month. And I'll show you what happened in these different treatments. And so if you put all species at the same time on day 0 and run this experiment for a month, then the the Mechanicorbia reucoffia and Gluconobacter species, one bacteria, one yeast species, are the ones that were dominant over time. And that made sense to us because actually these are the ones that we see much more dominant than the other two species. When you put the yeast species first, then this is what happened, that the all species except one went extinct, and it's only the yeast, one of the two yeast species, Mechanicorbia, that were just dominating and excluded all other species. And it's simply the mirror, the opposite, that if you then now put the bacteria first, then they were the one, the Gluconobacter was the one that dominated. And so when you get results like this, you don't really need to do statistical analysis because it's just so obvious that it's one of the clearest priority effect experiment that whichever species you put first are going to exclude others. And so I think that we have a strong priority effects here. But in California, we have a very strong daily fluctuation in temperature. So just out of curiosity, we also wanted to repeat this experiment under fluctuating temperature. And when we did that, mostly this was by Caroline Taka, who was doing the experiment. But when we did that, this is what we found with a simultaneous introduction. It's the same two species that were coexisting, but when you have the yeast species first, then the one of the bacteria, Gluconobacter, was barely there, and so there was no complete exclusion happening if you look at the middle panel. And then if you put the bacteria first, it's the other bacterial species was also just barely hanging there. And so in both cases, I think we have a strong priority effects, but relatively speaking, we can say that when you have a constant temperature, it's even stronger. And if you have a fluctuating temperature, it's weaker. And so here we can say that when temperature is fluctuating, then when temperature is constant, then we have a strong historical contingency happening. But obvious question is how to explain this priority. What could be the mechanism behind this particular priority effect? And so we've collected additional data to have some idea of what happened. And what I'm going to show you, this is just a conceptual diagram, but based on the data that we collected. And it turned out that it seems like the pH of nectar and the amino acid concentration are the two key factors that might explain priority effects between bacteria and yeast. And so we know that the yeast are good at keep growing in nectar, even when the amino acids are not so abundant. But they are not so good at growing when the pH gets low. On the other hand, if the bacteria, they themselves actually reduce pH, but they can continue to grow. But they're not so good at growing when amino acids are not so abundant. And on top of that, we also found that this is the change, that the chemical change that the yeast make, and this is the change that the bacteria make. So when they introduce in fresh nectar, yeast would reduce the amino acid concentration dramatically, but they don't change the nectar pH so much. And the opposite is true for bacteria. So you can already see what's happening, that it's not that complex. Basically, both of them are making nectar chemical environment more favorable to them, and making it very challenging for the other to come. And so it's the habitat modification that's happening that seems to lead to priority effects. And when you put these two species together, then this is the change that we can expect. And by the time they are making the environment not so good to both of them, they will move to new flower so they can sustain their population. At least that's what we think is happening. And this is under constant temperature. And under fluctuating temperature, we think that it's simply because everyone is a bit struggling, so that the growth rate is overall smaller, so that the extreme changes that happen to nectar chemistry doesn't happen, and so that that makes the coexistence easier to have. And so all of this just laboratory experiment. And so the fact that we got this result in the lab with a PCR tube and pipette doesn't necessarily mean that it's relevant in the field. And so we wanted to see what happens in the field, and this is just observational data, but we went to many flowers and just to observe, to measure the yeast density and bacterial density. So each of these dots represents a single flower. And as you can see, what we found, this is just observational data once again, but what we see is that if you have a flower with lots of yeast growing, then we don't see bacteria as much. And if you have, on the other hand, if you have a lot of bacteria growing in a flower, then we don't see yeast. So at least this observation is consistent with this idea that there's a strong antagonistic priority effects between yeast and bacteria. And so just to get back to this overarching question, when should history matter? In this case, it's just a simple, small experiment. But one potential answer to this question is that when the environment, the temperature, for example, is less variable, is when we expect history to be important. The next experiment that I wanted to talk about involves multiple species of yeast. So now we're just focusing on the yeast only, just ignoring bacteria for now. And some of them are closely related, some are not so closely related. And we did a similar kind of experiment. It's always simple, the priority effects experiment. You just want to flip the order and see what happens. And we also had monoculture as a kind of reference. And we did this with all possible pairs of species. It's just a pairwise interactions to start with, but we looked at all possible pairs. And so in this experiment, focusing on green species, this is one of the Meshonikovia species. I'll just show you what happens if you put this species first and then wait it for 2 days and introduce the other species that are on the x-axis. As long as you put this species first, there's no effect of the other species. But if you introduce this species second, then depending on which species you use, they're completely suppressed. And because we did all these experiments for all possible pairs, this is what we have. It's a little bit messy, but what you can see is that depending on which species you look at, there's some very strong effect of the specific identity of species of the other species that that may be competing, but it's a little bit messy, and we can't really find a systematic different way of explaining this, and so we're having a hard time. But we've thought that it could be interesting to think about this phylogenetic relatedness, because at the time when we did this experiment some time ago, this phylogenetic pattern became a little bit popular in community ecology. So we thought that even though nobody's we knew were looking at priority effects in relation to phylogenetic relatedness, we might be able to explain this variation in strengths of priority effects with the phylogenetic relatedness. And so priority effects can be either very strongly negative or strongly positive, facilitative. And when we plot our data, this is what we found. So that when we use closely related species, the priority effects were strongly negative. But if you have a distantly related species, the priority effects one's very strong. That doesn't necessarily mean that the competitive interactions were weak. The competition can be strongly affecting these distantly related species too, but presumably because when you have a distantly related species, they are ecologically very different, so that it doesn't really matter which order you use. There's always clearly competitively superior species. So even if that species come first or later, they always win. And so the history doesn't matter. On the other hand, if you have a closely related species, on average, there could be a lot of variation, but on average, these ecologically, they are very similar, so that the competitive race becomes sensitive to history. Whoever has a head start is going to win, which lead to strong priority effects. And so I'm not explaining so much ecologically what exactly happened, but briefly we can say that another potential answer you can give to this question, when should history matter, is that when species are closely related, you would expect the history to be important. The next experiment that I wanted to briefly talk about, it's getting brief, more brief, my explanation of these different experiments. But here, as an ecologist, we weren't completely satisfied with just talking about phylogenetic relatedness. We wanted to actually directly look at ecological differences among species. And so what we decided to do with Rachel Vanette, who is now a professor at UC Davis, at that time, she was a postdoc with me. We measured lots of things about each species of nectar yeast, each different species of nectar yeast, in terms of how good they are in growing when they are given specific kinds of amino acids or sugar, or how much change they make to the hydrogen peroxide in nectar, or the amino acids and sugar, and all these different things, and also how similar they are. So when we collected all of these, what we thought was ecologically relevant data, we won't be able to explain the strengths or priority effects if we put all of these together. For example, if you just do PCA to summarize these different ecological data, we weren't having a good explanation. Only when we thought carefully about what these different data mean, does that mean this is about resource use, this is about more of a what they do to the, rather than their response to the chemistry, but it's more about their impact on the environment. And so we try to sort out different data into what we call niche overlap, impact niche, and requirement niche. When we did that, we started to have some systematic explanation, so that if you, for example, if you look at panel A, when this is not surprising, it's kind of all expected, but if you have a high overlap with the ecological, if species are very similar, then it leads to very strong negative priority effects, and so on. And so overall, if we put all these together, about 60% of our variation in this also laboratory experiment, in terms of the strengths of priority effects, we were able to explain with this niche related data. And so getting back to this quickly, another thing you can say is that you can actually measure different things about related to the species ecological niche to have some good prediction, reasonable prediction from ecological standards in terms of when history should matter, which set of species should show some strong priority effects. The next experiment I wanted to briefly talk about involves application of what's called modern coexistence theory. In community ecology, this is an ongoing popular line of research to try to apply Peter Chesson's idea of using what he calls fitness difference and stabilizing difference. And you can estimate how different species are and how much of a intraspecific versus interspecific effect different species have, and that's related to stabilizing difference. Without giving you the details of this theory, I wanted to just graphically show here that you can actually map the species pair into this region where if you have a strong stabilizing difference and then small fitness difference, then you expect coexistence of species. And if the two species are very different in fitness, then so that the fitness difference is high, then you expect competitive exclusion. And then you have a negative stabilizing difference and then small fitness difference, you expect priority effects. And so the Tess Granger, who came from University of Toronto to do this experiment with us with nectar yeast, she also looked at different pairs of species of nectar yeast and under different conditions. And when she did an experiment, you can plot these different pairs into this region to without measuring so much about niches, just observing how well they're growing, you have some predictions of where these different pairs should go in this region. And so what we did is to see whether the prediction coming from this modern coexistence theory can actually explain actual priority effects. And so this takes a little bit of explanation, but on x-axis, I have a predicted strengths of priority effects, and on y-axis, I have observed strengths of priority effects. And so on y-axis, let's say, let me start with the x-axis, the distance to exclusion priority effects border. If I go back to this figure, if you have a dot, that's well within the priority effect. So if you have this border between competitive exclusion and priority effects, if you have a dot that's very far distant from that borderline, competitive exclusion and priority effects, I would expect that there's a strong priority effect. On the other hand, if you have a dot, the pair that's sitting very close to this border between exclusion and priority effect, then priority effect may be weaker. And so that's what I'm plotting on x-axis here. And then on y-axis, what Tess did experimentally is to, when she had two species potentially competing, when the second species came in this usual priority effect experiment, in addition to the second species, she also added lots of amino acids, assuming that that's the limiting resource, and see whether we can save the second species by giving them lots of resources so that they can cope with the other species that might have already affected amino acid concentration. And what we see is that there's a good relationship between these two. There's not many replicates here, but the prediction that you can get this theory seems to actually explain the actual priority effects. And so this is getting a little bit superficial, but I wanted to say that when this answer to this question, when should history matter, can also come from modern coexistence theory. That's implicit about, this theory is a little bit implicit about the ecological characteristics. But even so, you can think about fitness difference and stabilized differences, if this is your favorite theory, to explain when to expect strong priority effects. I think I have two more experiments to show you, and then I want to start to wrap up. But here, this is unpublished results, but I thought it was interesting to share with you. This is an experiment done with my current PhD student, Kelly Chappell. She was interested in evolutionary question. And so what she did was an experiment where we had this isolate, one isolate of Mechanicorbia ricoffii, Nectar is we isolated from the real flower in the field, and we call it ancestor for lack of better term. And then in artificial nectar in the lab, she was doing just experimental dispersal every other day to simulate what might be happening in the field. And this was repeated 30 times, which led to about 2 months of simple transfer of nectar with this yeast in the lab, and expecting that potentially, because they have such a short generation time, potentially, eventually there could be some evolution that could happen. And we did this experiment because I think this 2 months corresponds to roughly the flowering season that we have. And so this monkey flower, they bloom in spring, and it lasts, at least at a local field site, the field flowering season lasts for about few months, 2 months or so. And so our interest was, can we expect any kind of rapid evolution that could lead to some changes in priority effects, even within that single flowering season? And so as kind of preparation for eventual priority effect experiment, she prepared these 3 different treatments: the ancestor, the isolates that we collected from the field and stored in the freezer, and we also had the strains that we had by doing this serials transfer in a what we call fresh nectar, nice nutrient-rich nectar. We had two other treatments where instead it's the same with a fresh nectar treatment, with the exception of this nectar in one of the treatments we had a very low pH to try to simulate what might happen if bacteria were already there. And then we also had maybe more realistic simulation of what could happen if you have a lot of bacteria in the region. We also had the nectar, the same artificial nectar, but previously conditioned by growing bacteria and then filtered out the bacteria. So it was the bacteria conditioned nectar. And getting back to priority effects, what we did is then use these different strains that we've prepared and stored in the freezer to see whether we see any differences in terms of the strengths of priority effects. So you can do the usual experiment with bacteria, the yeast, the Messinicoria leucophila that we have now, and then we had Acinetobacter, another common bacterial species that we see in nectar, and Kelly was doing experiments, priority effects experiment between this nectar yeast and then bacteria, and then measured the strengths of priority effects. And this is the results that we have. A little bit subtle, but we do see some statistical difference, so that the ancestral, what we call ancestral isolate strain that we have show negative priority effect as expected, that if they came late in relation to the bacteria, they suffer because of this change in pH presumably, and so there's a negative priority effects. But when you have the strains that have been growing in low pH constantly over 2 months, so I'm now looking at the low pH nectar, then there's a statistically significant difference such that for some reason this strain that were growing in low pH nectar doesn't suffer strong negative priority effects, relatively speaking, compared to ancestral one. And that's the same with the bacteria conditioned nectar. But we don't see that difference between ancestral and involved if we look at normal nectar. And so it's not completely surprising, but it's still a kind of nice experimental demonstration that even with just a few months of potential evolution, if they are constantly challenged by having to cope with the potential competitor, bacteria in this case, eventually you can expect some evolution of resistance to priority effects, so that the priority effects get weaker as a result. One kind of secondary note that I wanted to share is that I'm here in the left panel, I'm showing part of the figure I already showed you in the previous slide, this one, that the normal, the bacteria that are growing in normal nectar show stronger negative priority effects compared to the low pH treatment. So that's just, I'm just showing the same thing. But if I look at how well they grow in monoculture, so no bacteria, but they're just growing in nectar, then the strain that were conditioned, that were growing in normal nectar, has a faster growth in this nectar, which is also not that surprising. I think it makes sense that if they are being grown and potentially adapted to this particular normal nectar, what we call normal nectar, they might be more adapted to grow fast in that environment compared to the low pH one. But what we see, what we think is potentially interesting, is that there could be a trade-off between how good you can be in resisting priority effects and how well they can grow if you are just by yourself. And so there might be some interesting trade-off going on here. But getting back to this question once again, this particular experiment, I think, is suggesting that the history can be more important when species are evolutionarily naive in relation to the competition and interactions that they might have that result in priority effects. The last experiment that I want to briefly talk about is a field experiment, and this is sort of an indirect evidence, not so much direct evidence for in terms of priority effects. But we did this simple experiment where we had bagged flowers and caged flowers where only hummingbirds are excluded and not the bees that also occasionally come to our flowers, and then control exposed flowers. And what I'm showing here is DNA sequence data that characterize bacterial species composition, and each dot represents a flower. And so what I hope you can see is that if you look at blue dots that represent bagged flowers, there are more, there's still a lot of variation spread in this ordination plot, but relatively speaking, the blue dots are more tightly clustered compared to particularly the orange, the exposed flowers. And so that there's more variation in species composition among exposed flowers compared to bagged flowers where the pollinators couldn't come. And then the gray dots are showing the data that we have from hummingbirds. And so in this, the same field where we did this field experiment, we also caught by using MistNet as many hummingbirds as possible. And then we make the birds drink clean sugar water, and then the remaining sugar water we used to try to analyze what they were carrying in their tongue and beak. And so each dot here, the gray dot, represents an individual hummingbird. And what I hope you can see is that if you look at exposed flowers, some of them are kind of pulled toward the direction of what hummingbirds have, but the hummingbirds themselves are also variable because there are some gray dots that are up in this panel and then and below. And so some exposed flowers are being kind of pulled towards particular hummingbirds, and others are going toward the other one, and that doesn't happen as much in bagged flowers. And you can quantify that difference in variation among different flowers in terms of species composition, but by quantifying beta diversity. And this is just a confirmation of the pattern that I was trying to describe, that if you look at exposed flowers, the beta diversity is higher than bagged or caged flowers. And so this is all data I have, but the one speculation you can make is that if you have hummingbirds coming to introduce different species that vary among individual hummingbirds, then because they introduce variation in immigration history, so that lead to potential priority effects. So the potential for priority effects producing variation among flowers are there, but you have to have the variation, actual variation in immigration history introduced to the system to see the effect of priority effects. And so this is once again sort of a speculation, but another potential answer you can give to this question is that when immigration is frequent, so that many species come and leading to variation in immigration history, you have to have that variation in the first place to see a historical effect. Okay, so I think I have five more minutes or so. So I have two additional stories that I wanted to add to the experiment I already talked about to try to convince you that this peculiar communities, to study them can be interesting, even if you're not so interested in nectar microbial communities in and of themselves. And so I've been focusing on this picture here, that there could be a priority effects between yeast and bacteria, and I showed you priority effect within yeast also, and I think we also have some priority effects in bacteria too. But so this could be interesting if you are interested in, like Robert Schaefer and his group at Utah State. If you are interested in nectar microbes, this could be interesting. But if you are a plant ecologist, does this really matter? The microbial ecologists can do these experiments, but but we can let them do that, and we don't really need to think about these tiny communities that are happening in Nectar. And so I wanted to question that, and so we've been doing experiments, and I think Robert has done some really nice work also, and as well as others that are related to this. But is it really true that these two things are separate? We know that the microbes are dependent on plant and pollinators. As I was explaining, it's obvious that the plant is needed for the yeast and bacteria because they provide nectar. So that's the resource that the habitat they use. And then the pollinators are the ones that help them disperse among flower to flower. And so we know that the nectar microbes are dependent on these larger organisms, but is it possible that the microbes themselves then in turn affect plant pollinator mutualism? And it turned out that through the series of experiments that I'm not going to, in the interest of time, I won't be able to share actual results, but we were seeing is that what we saw is that because of the strong changes in nectar chemistry that are possible with microbial growth, they can have actually effect on plant pollinator mutualism, so that if you can experimentally introduce bacteria to actual flowers, and what we saw is that the pollinators don't come to bacteria colonize flower as much, and also when we look at the pollination success and in seed set, we see reduced success of plant reproduction if you introduce bacteria. On the other hand, even though yeast also changed nectar chemistry very much, we don't see, if we do the same kind of experiment where experimentally introduce yeast, we don't see any detectable effect of the yeast introduction, experimental introduction on either pollinator preference or seed production. And so that's why I'm putting that arrow as a dotted arrow. And what's interesting here is that if that's the case, the priority effects that happen within nectar microbial community could have an effect on plant pollinator mutualism. For example, if you have yeast dominating because of priority effects, and they are the ones that came early, and then suppressing bacteria, that could have an indirect effect, indirectly facilitating plant pollinator mutualism because they suppress the bacteria that might degrade the mutualistic relationship here. And so even if you are a hummingbird biologist or plant ecologist, this could be an interesting topic to think about. And then the second thing is that I was always bothered by this fact that this community is so tiny that in the large scheme of things with a large spatial context of ecology with other organisms playing a role in ecosystem, does this really matter if you just studying this tiny system? And so what we decided to do is to go out in the field, and then for the whole plant, we introduced yeast in all opening flowers in a plant, and we had another replicate treatment where we introduced bacteria to all flowers that are blooming, and then we have water as a control to see in addition to what's happening at the single flower scale, do we have some larger scale phenomena where priority effects can happen at the larger scale? And so what we found is that after that introduction, we went back to these plants that are replicated and then sampled nectar from opening flower at that time. So because each individual flower lasts for about a week, roughly speaking, if you went back in 3 weeks after this experimental introduction, you are looking at what happened 3 generations of flowers after your introduction. And even so, the flowers that we were sampling the nectar from is not the same as the one that we did this inoculation. But even so, if you look at the bacterial species and fungal species composition, we saw a difference, meaning that it's not just the individual flower where priority effects happen, but potentially this experiment kind of suggests, what this experiment suggests is that once a plant becomes dominated by yeast, for example, then they can stay like that because of the local priority effects that happen within flower and local spread of local dispersal of these yeast that are now dominant within the plant. The plant might stay yeast dominated, yeast colonized. And so even at the larger scale, than the single flower level, we might have implications, the consequences of local priority effects. And so I have to wrap up now. I think I might have been trying to put too much into one talk, but I wanted to show you what you can do to ask this question: when should history matter? These are the answers, potential answers that I gave. And when you just focus on this slide only, I have nothing here about nectar microbes. And so my hope, even though I don't know how well we are achieving this, but our hope is that by using this nectar microbes, in addition to understanding the microbial communities there, we could have potentially general message saying that these are the kind of things that we can apply to different systems to have some, try to cumulatively, incrementally lead to some generally applicable ideas about when should history matter. Okay, I think that's the end of my talk, and I hope that we have some time for Q&A. Thank you very much.